Hola, buenas tardes a todos, a todos. Os voy a presentar a Mireia Mestre, que es una recién incorporada al Departamento de Biogeoquímica y Ecología Microbiana del Museo, con un Ramón y Cajal. Y que yo estoy encantada de presentarla porque creo que compartimos dos de nuestras pasiones, que es el estudio de los microorganismos, de aquello que no vemos a simple vista, y también el estudio de los ambientes polares, sobre todo de la Antártida. Entonces, pues con ella me unen estas cosas. Entonces, Mireia, ella se define como ecóloga, pero yo creo que tiene también mucho de microbióloga. Y ella tiene una licenciatura en biología y luego tiene una segunda licenciatura en ciencias del mar, que la hizo en la Universidad de Alicante, y posteriormente hizo el máster en ciencias ambientales en la especialidad de ecología. Realizó su tesis doctoral en el Instituto de Ciencias del Mar en Barcelona, centrándose en el estudio de la diversidad y la función del microbioma marino antártico. Durante su tesis doctoral hizo bastantes publicaciones con mucha relevancia y también recibió el premio Landmark Paper Award en el 2018 y tiene el premio extraordinario en la licenciatura de Ciencias del Mar, en esta segunda licenciatura, y también es premio extraordinario de tesis doctoral. Posteriormente tiene una etapa postdoctoral en Chile de ¿cuántos años? Casi 5 años en Chile, primero en la Universidad Austral en Valdivia y posteriormente en la Universidad de Concepción. Y allí ya ha liderado su propia línea de investigación, que está centrada sobre todo en el estudio de la diversidad y función del microbioma marino antártico, concretamente el microbioma asociado al zooplancton antártico. Ahora ya se ha incorporado al museo, como os decía, con la intención de seguir con esta línea del estudio de las comunidades microbianas, sobre todo asociadas a otros organismos en la Antártida, pero también pretende desarrollar una nueva línea de investigación que ya ha empezado con ella un poco más de forma teórica y de la cual nos va a hablar ella, con lo cual lo va a explicar ella mucho mejor que yo y prefiero darle paso a ella. Entonces, cuando quieras.
Hola a todos. Hello, everyone. Thank you for coming here today. Today I'm going to talk in this seminar about the Microbial Conveyor Belt. That is a theory that we developed like 2 years ago. And this will be the outline of the talk. First, I will start to talk a little bit about myself. Then you can know me a little bit more. And then I will talk about the Microbial Conveyor Belt. This is quite long, and the 6 sections are mostly the same sections that we can find in the paper. So we can screen all the paper generally. And finally, I will explain some examples of how can we apply this theory to the museum. Okay, I like to start my talks with this view of the biosphere. As you know, this is the only known planet that has life, and this life can be macroscopic and microscopic. I'm originally from the Mediterranean Sea, the eastern coast of Spain, and but the truth is that half of my life I live in other places, and even though my family is still there. So when I was a kid, I was living surrounded by nature, so it's not surprising that I ended up studying natural science. First, I did a bachelor in biology. After that, a bachelor in oceanography. And when I finished the bachelor's, I did my master in environmental science. And after that, I did my PhD in oceanography, where I focus on microbiology. Okay. These things are somehow linked because in biology I specialized in genetics, in the Bachelor of Oceanography in marine ecology. In environmental science master, I have like an overview of all this. And finally, during my PhD, I could apply all this knowledge to this line of microbial oceanography. So To talk about a little bit about my PhD, this was focused, as I said, on the ocean microbiome, and I studied how the diversity of microbes vary, spatial and temporary. Okay. For this, I focus on the micro scale and the macro scale. The micro scale were the marine particles, where they have a lot of marine biodiversity, and the largest scale was the global ocean. So for this, I had samples from the global ocean and also from the Mediterranean Sea. And the interesting thing of the Mediterranean sample is that this was a time series where I could describe the diversity, how the diversity vary along time in this station. From the PhD, the most outstanding results were that we find a connectivity between the surface and the deep ocean microbial communities, and that this connectivity is very high and is thanks to the sinking particles. In the ocean, we have a lot of organic matter that is produced in the surface, and this eventually goes to the deep ocean. So those particles carry microbes that transport those microorganisms to the deep ocean. So after that, I did my postdoc. This was in Chile, and here I focus on Antarctic ecosystems. Particularly, I focus on the krill microbiome. Here I studied the gut microbiome and the skin microbiome and the microbiome linked to fecal pellets. And I linked this to the microbes present in the water column. The interesting part of this stage for my PhD is like I could become like an independent scientist, because I won 3 projects where I was the principal investigator. So I learned how to manage projects during the pandemics. That was quite difficult. And also how to teach students. I had bachelor and master students. And now, this stage is now, as a Ramón y Cajal researcher, I'm here in the museum, and this is my lab. I call it the M lab, and the M, as you can see, can stand for microbiomes and macroecology, but also is related to my name. So there are many M's around, so I decided to call it the M lab. Okay, and now I want to develop more about this theory, but first I want to explain you in certain detail what is this theory, how this was built. And for this, we start from the beginning. I know there are not many microbiologists here, so this introduction will be very general. Microbes are very important for the biosphere because they are highly diverse. They have, even though they are very small and we cannot see them, they are a great part of the biomass of the planet. And they also have very different functions to help the biosphere in different ways. For example, in the biogeochemical cycles. But there are many things from the microbes that we still don't know. For example, we don't know if the distribution globally is endemic or they are everywhere. This means they are ubiquitous. So, and this leads us to a famous sentence in microbiology that says everything is everywhere. This sentence, even though it has like a century, is still, we still don't have a clear answer about it. But then we thought, if a microorganism is able to be everywhere, needs two things. One, has to be able to disperse very long distances and has to be a strategy of dormancy. Then this microbe can survive environments that are not suitable all around this travel. And this, I think, sounds quite obvious, but we realize that this is not written in any paper. So we conclude that maybe it will be nice and interesting to build more theories to understand the microbial diversity around the world. And of course, we were not the first one realizing about it. Because, for example, in this paper from 2020, entitled Conceptual Challenges in Microbial Community Ecology, Prosen and Martinez said that microbiology needs to include new concepts and theories to improve our understanding of microbial ecology. So this encourages us to build a theory. And how do we build this theory? For building our theory, the Microbial Conveyor Belt, we made this path. We use node data that is not quite common in science to use node data, but we use just only bibliography. And for this, our tools were logic and deduction and an holistic approach. And after all this trip, we were able to construct this theory, the Microbial Conveyor Belt. So now I'm going to explain this theory. And for this, I will start talking about the building bricks of this theory. The building bricks of the theory is the microbial seed bank cycle. This microbial seed bank cycle, for constructing this picture, we searched for information about the dormancy and activation of microbes and how they change from rare to abundant in the nature. And we saw that microbes change from suitable environment to unsuitable environment, and then in the suitable environment, they grow and concentrate, and when they are moved to an environment that is not suitable, they become dormant, they dilute, and they become dead. So this is a cycle. That's why it's the microbial seed bank cycle. And this cycle has two dimensions, the spatial and temporal. The temporal is, for example, a microbe that doesn't move from the place, but somehow waits for the suitable environment. So when this is, for example, seasonality. Every time, for example, this microbe likes summer, so every summer will be a cycle. This is the temporal dimension. But in the spatial dimension, we will have a microbe that will move from a suitable environment to an unsuitable, or the other way around. Okay? And in this talk, and for this theory, we will focus especially in the temporal transition. Okay. And these spatial transitions of the dispersion, we saw and we realized that has three interesting things. The first is that the bibliography says that the microbes have specific adaptations to the unsuitable environments. This means that if they are in anoxia, they have a specific mechanism for it. If they try to avoid high temperatures, they have also in the cellular mechanisms to avoid it, etc. There are many examples of that. And we also see that microbes are able to become dormant even if the conditions are suitable. And this means that those microorganisms know that the conditions are changing and they are getting ready for it, okay? So they doesn't wait to be in an environment that doesn't suit them. They know this, they sense this in advance, and they become dormant. And in the other direction, we saw in the literature that those microorganisms are able to sense that they are arriving to a place that is suitable, and they become active. This is, for example, in rivers, where they in the river, they become active just before, in the path through the lake. If their suitable environment is the lake, they will become active in this river. And what all this means? This means that microbes are adapted to this dispersion. And then we can conclude that the dispersion is not random. The dispersion of microorganisms is normally considered as random, but after putting all this knowledge together, we can conclude that at least in some occasions, this is not random. Okay, and how is this possible? This means for this being possible, we need that those traits are selected. And the only way to select those traits is because eventually one organism goes back to their origin. So after the dispersion, they are selected and they are back. Okay, this means that what I say that they are selected like this, and the only way is that they are back. And then we can conclude that the dispersion of microorganisms can be spatially cyclical because the origin is also the end, and this recurrent. This means that this loop is repeated over time. And we can add more information to this, and we saw that this dispersion can be global. And we conclude this after seeing that there are papers that say that the dispersion of microorganisms to long distances, for example, the dispersion of microorganisms through the atmosphere, has also seasonality. So this has like a recurrency. Okay? So yes. This is basically the theory that we say the dispersion is spatially cyclical, is recurrent, and is global. And this is the microbial conveyor belt. But this is not that simple. So there is not one event of dispersion for moving this microbe all around the world. We will see many events that are connected between them, and during these different events of dispersion, the microbe will go through different ecosystems. Okay? For example, they can move from the land to aquatic ecosystems, or from the atmosphere to the land, etc. So I think maybe this is more useful for understanding this. Here we see conceptually this flux of microbes around the world, and they connect different air compartments, for example, atmosphere, lithosphere, and hydrosphere. And they also connect the microbial communities around the world. Okay, and now if we add some complexity to this, we will see that the global microbial conveyor belt is formed with partial conveyor belts. The partial conveyor belts are exactly the bricks, the microbial sequence cycles. And here we have two examples of it. For example, this is in the water column, how they cycle from suitable environment. This is, for example, photosynthetic microorganisms to an environment that is not suitable. Okay, and a quite similar example is this, in which the microbe goes to a place that is not suitable when they are ingested by, for example, the krill. So the intestinal tract is not okay for them, and they become dormant, and they will be released again with the fecal pellets, and then they have again the opportunity to thrive. Okay, those two examples of microbial sequence cycles are somehow connected because a microorganism can be here, for example, in the upper part of the ocean, can do this loop, or maybe can be eaten by a krill and then do another loop. So they are connected. Then we see that from the sequence cycle, we can have a meta-cycle of cycles connected among them, and all of them connected will be the microbial conveyor belt at a global scale. So summing up, we have the microbial sequence cycles here, and the microbial conveyor belt will be all these together. It's okay? Yeah, okay. So, I think I'm repeating this again. This connects microbial communities, air compartments, ecosystem, environments, etc. And we have here different examples at different scales. Global scale, this is a smaller scale connecting atmosphere, lithosphere, and the ocean. And this is a particular loop in the ocean that I will explain later more in detail. This belt is closely linked also to biogeochemical cycles. Here we can see, for example, this is the carbon cycle, and this is the hydrological cycle, and we can see that they are, no, always. This is the carbon cycle, and this is the hydrological cycle, and we can see the similarities. So if the microbial conveyor belt transports the microbes that travels around the world, no, if the biogeochemical cycles moves the elements through the earth cyclically, then the microbial conveyor belt what do is to transport those microorganisms that are in part responsible of these biogeochemical cycles. So here we move the elements, and here we move the bioreactors that are the microbes that will transform the element. Okay, and how can we outline the microbial conveyor belt, or the many of them, or some of them? One idea is to define first key species in an ecosystem, for example, photosynthetic ones, and then we have to see which dispersion avenues those have. For example, if they are dispersed through the atmosphere, or through a river, or through the ocean currents. Okay, we have to know which vectors participate. There can be the wind, can be the water, can be some animals, for example, carrying them during the migration. Okay. And then after all this, we have to build the loop that is the microbial sequence cycle. That is the partial conveyor belt, and then we'll know this contribution will be a part of the global microbial conveyor belt. Okay, this is an example that we built following those ideas, but we built not for When taking samples, we read it with the information from the papers. And yes, we linked this cycle with the carbon fluxes. We see here the darker arrow is the seed-bound cycle, and the dotted one is the carbon fluxes. And we see they go together. So all these, the papers tell us that microbes grow in the suitable conditions, the phototrophs, for example, in the upper part of the ocean, and then they will sink through the gravitational pump, this huge transport of carbon from the surface of the ocean to the deep ocean. Okay, and those carry the microbes through the deep ocean, and especially those that are photosynthetic will become dormant in the deep ocean. And when they are here, they will get diluted with the lateral transport of the deep water masses. And this big transport along the oceans will drive microbes at certain point. They will have the opportunity to go again to the surface, for example, through the upwellings. And here the microbes will become active again because they will reach the suitable part. For them, they will be again in contact with the light, okay? And the loop begins again. This is strongly linked to the carbon cycle, as you can see here, and also to the ocean circulation. And here, as this is the carbon fluxes, as I said, so this is potentially also a link of this cycle to another connected to the atmosphere. Okay, for example, in the ocean, when the water is evaporated, it's carrying a lot of microbes. Okay, so this is a partial one, but can be connected to many others. Can be connected to the one of the organisms eating these microbes, can be connected to those microorganisms going to the atmosphere, etc. Or also sinking to the bottom. So this theory has many implications for microbes, for other organisms, for the planet Earth, and for the humans. Why? Because for microorganisms, define where they thrive or not, if their conditions are suitable or not, and they also has a strong effect in their selection. Other organisms, this is quite related on how they evolve. Now we know that microbes that carry the host are related to how they grow, how they thrive, the digestion, the health, etc. There are many links. This is another topic, quite related. And for the planet Earth has many implications because it is strongly linked to the biogeochemical cycles, has an impact in the ecosystems because they move the microbes that participate on those ecosystems, and also has a role in the evolution of Earth system because if the Earth system evolved with the biogeochemical cycles, the microbes also participate in this evolution. And for us, of course, this is important because we live in a world that is full of microbes, and those microbes directly or indirectly give us ecosystem services. But without them, we will get extinct very rapidly. Okay, this has also implications for other things related to microbiology. For example, this is a famous picture, yes. In microbiology, that we have this is the Rankine curve, where we have ordered the different microorganisms in a sample from the more abundant to the less abundant. And one particular thing from microbes is that we always see a very long tail of rare microorganisms. We still don't understand why this is so highly diverse. And the microbial conveyor belt can help to understand this high diversity because microbes are circulating continuously through the different communities and ecosystems. So this ensures that the flux is continuous and they are entering all the time, and then contributing to this diversity of the rare biosphere. What can help the microbial conveyor belt can also help us to understand, for example, the population dynamics of some microbes. For example, we can know where they are and how they thrive or not in the environment. But this also contributes to know not only the populations, but also the community dynamics, because it can help us to understand how the different communities are connected between them. And of course, as we say, the theories of the global connections in the planet. Okay, but But this belt has problems. And the main problem we detect is the global change. The global change can transform this global flow of microbes in many, many ways. For example, a climate change, particularly inside the global change, can or is changing the intensity and the direction of currents in the wind, or for example, in the water current. So then they are breaking this loop. Okay, they are also changing the evaporation rates in the ocean. Another example can be, for example, when we construct a harbor, we are destroying also this flow. Or if we build dams, we are also destroying the loop of microbes in the rivers. Or if the river becomes dry, we will never have this flow of microbes again going from the atmosphere to the ocean in this particular place. Another threat related to the global change is, for example, if we introduce new vectors to the ecosystem. This can be, for example, the plastics in the oceans that can carry pathogens or microbes from one place to another. And this is also a problem when we introduce invasive species to a new ecosystem, because those invasive species carry many microorganisms, some of them pathogens that can affect others. Or maybe they are carrying some microorganisms that were not part of the new ecosystem, but then they are released them and do transform all the local communities of microbes. So we can say that if we disturb the microbial conveyor belt, we will transform the diversity of the ecosystems, and then the ecosystem services will be damaged. And we also will promote the dispersion of pathogens. This part can be linked to, for example, the COVID pandemics. But we can talk about this another day. Okay, so for concluding, we need conceptual frameworks to understand better microbial ecology. We are generating a lot of data, but maybe we have to really think about what this data means, or what all the publications together are telling us. And we can highlight also that the microbial conveyor belt help us to understand the links between dormancy and dispersion, the links between biogeography and ubiquity of microbes, also the connectivity between ecosystems and the resilience. and the links between microbes and biogeochemical cycles. So in the future, I propose that two things. That for disentangle the microbial conveyor belt, we will need a collaborative effort between disciplines. For example, microbiologists working with people, with experts of the atmospheric circulation, for example, or experts with some vectors that are, for example, animals or migrant animals. Okay. And we think we should take on account this flux of microbes in management plans in order to protect this conveyor belt and this flux. We need this flux to be present because this helps us to have more healthy ecosystems. Okay, and healthy ecosystems will give us ecosystem services. Okay, so finally, this is the paper, the publication that I say was published in June in 2021 in Trends in Microbiology. This is you already know this picture that was in the cover of the journal. And this was made with two people, myself and Juan Hofer. He's a scientist in Chile. Okay. And of course, I have to give thanks to all the institutions to support us all this time when we were developing the theory. This is the Centro Ideal de la Universidad Austral de Chile, la Agencia Nacional de Investigación y Desarrollo de Chile, INACH, that is the Instituto Antártico Chileno, la AWR Funding, que es the Antarctic Wildlife Research Fund, a la Universidad de Concepción y a la Pontificia Universidad Católica de Valparaíso. Okay, now this is the last part of the talk, and I wanted to show you how we can link this theory to the work we do in the museums. So this theory can be applied in many environments and many topics, but I wanted to show this particular one because I think it's of general interest here. So I will start with this example. This paper was published very recently in 2022, and is entitled "Gut Microbiome of Century-Old Snail Specimens Stable Across Time in Preservation." In this paper, they work with Oreolix atrigosa, ¿cómo era? Oreolix strigosa, this snail lives in the western part of the United States and lives in very different environments. You can find this snail living in the grass, for example, in grasslands, but also in the Rocky Mountains. So it's quite abundant. The habitat range is huge, and in this big area, they can live in very different parts: grass, sand, mountains, etc. Okay. So what they did, they had samples of a time range of 100 years. Exactly is 98 years, but they had samples like from this last century of this snail. So they extracted the DNA of those samples that were preserved in ethanol. And they could see if the microbiome changes over time. Okay? So they see that over time, the last century, the microbiome was quite conserved. It doesn't really change, but what they could see that the major driver of the differences in the community composition was the locality. Okay, so all these trees were conserved the last century. There is not a real impact on this. And I think the take-home message that at least inspired me is the last sentence of the paper that say, this study should encourage further use of museums as a resource for studying microbial ecology across time and space. Okay. And the second example I have is one that was done here. Two scientists from this museum participate, Patricia and Ignacio. And they published this paper entitled Detection of the Amphibian Chytrid fungus Batrachochytrium dendrobatidis in museum specimens of Andean aquatic birds, implications for pathogen dispersal. What they did here is they took samples from two collections of museums, one from the Colección Boliviana de la Fauna in Bolivia, and also samples from the Estación Biológica de Doñana. And what they did here is they took samples from the legs of the ducks, and they discovered that this chytrid was present there. And why is this important? I forgot to say this chytrid is really important because it's the responsible of the decline, the extinction of many amphibian species and also the decline of many populations. So this is a very serious global problem. And what they saw here in this study is that with the samples from the museum, they can detect the vector, one of the vectors of this pathogen. And after this work, they did another related one, now with samples from many other museums. I think there were 3 museums from Bolivia, 2 from Spain, and 3 from the United States. And here they took samples not only from birds, also from frogs. And with all this data, they were able to describe the spatial distribution of this pathogen in the Bolivian Andes. And they could relate this distribution to the decline of the amphibian species. So those samples were from the 80s, and the decline was on the 80s, and they could match the distribution of this fungi with where the population declined. Okay, I think this is quite interesting of how many things we can do with samples from museums if we study their microbiome. So we are close to the end, and yes, I'm sure that there are many samples in the museums that are waiting someone to analyze their microbiome. So if any scientist around is engaged with this topic, I will be very happy to talk with these scientists, especially if they are related to the collections, so we can collaborate. So for this, we have some cards here. You can take them with my contact. You can copy the contact from here, but the cards are free for everyone. I think, yes, I tried to make them beautiful so you can keep them for a long time. So thank you very much for coming, for listening this topic. And yeah, this is almost everything. Now I'm trying to find also students. So if there are some students around, or you know some students that wants to be involved in the studies of clear microbiome, of the belt or museum microbiome, please tell me. That's all.
Thanks, Mireia, for this interesting talk. So now we have time for questions. So I will let that first. Someone from there ask. Yeah, you have already one.
Hi, Mireia. Congratulations on the talk. Really interesting. I was just wondering, because you said that you have a background in genetics. You're doing genetics, and you didn't say anything about. I mean, in this theory, there is no. You didn't mention anything about proving this, getting some proofs about microbes being, for instance, in photosynthetic microbes, being in shallow waters and then in the deep waters. Have you found any signal of that in paper? Because I guess if you screen, if your theory is true, you should find them, right?
Yes,
In the
there
bottom.
are many proofs, and exactly this match with my PhD. Because the truth is that during my PhD, we detected this high connection between the surface microorganisms and the deep ocean microorganisms. And we could see that the same OTUs that were in the surface were in the deep water. And this was the truly beginning of this theory, because I was thinking a lot of how it's possible that this happens, that if this happens always and they are selected to become dormant here, they have to somehow return to the surface. So yes, we have the evidences from the literature, the evidences from the data from my thesis, and also the links with all this.
Can I challenge this again?
Yes.
Did you take samples, deep water samples, and then put them to grow at the very, on the surface, so that to get to know if they are really dormant? Because you can find DNA in deep waters, and they can be eDNA that can be from animals or from microbes that are already dead. But how do you prove? How can you test that again?
Yes, this is also very interesting. I was not involved with this, but in the Instituto de Ciencias del Mar, after my thesis, they did some. Of course, these are big projects, and there are many people involved in that. And some scientists took samples from the deep water, and they did this kind of experiments. Okay, they did different treatments with light and that, and they discovered that many dormant microorganisms in the deep oceans become active when they grow in surface conditions.
I'm convinced.
Yes.
Hi, Mireia. Thank you for the talk. I was wondering if during the conceptual framework, have you considered reservoirs that become apart from the conveyor belt? Because, of course, the conveyor belt is really related to the ocean, to the atmosphere, but I was thinking about subsurface reservoirs and how they match in the theory, and if the theory considers that during the time, I guess millions of years, microbes differentiate or not. I mean, if you have any hint about that.
Yes, of course. First, the reservoirs are part of the belt also, but this is more related with the temporal dimension. Okay, so they are there, and eventually they are released again. And there are publications that are that include sitting gas, and like an example, and they study the microbes here in the deep in the in the deep ocean, no, in the subsurface ocean, and they linked this to the fluxes of microbes in the water column and also there, and they say yes, they are a reservoir, and also eventually during now, but during many ages, because this can be very ancient, microbes are released again to the water column.
Do you know if these microbes are similar to some, or I mean, what is the genetic difference with those that are already in the system, or if they differentiate over time?
Yes,
or
they send some match, but of course there are many of them that were not released before, and they are being released now. And another example to this is, for example, what happened with all those microbes that are reservoirs that are being released now because the climate change. For example, those with the permafrost or with the glaciers. And we already know that in some cases there are pathogens that are released that were, for example, not seen in many years, and now they are again in the ecosystem. And maybe Carles can talk about this, but I don't want. Okay, he has a nice paper that it's entitled Time Travel of Microorganisms, and talk about this kind of reservoirs.
He will talk about that in the jornadas,
Yes.
actually.
Yes.
Thank you. My name is Ana Martinez. I am from Mexico and I am a chemist. So sorry if I made some questions that are out of the scope. But you mentioned something about the plastics, and
Yes.
I am wondering if you know something about the microplastics and nanoplastics. Because when I see this cycle, I am studying that. That's why I
Yes.
ask you. When I saw this cycle, you can find micro and nanoplastic all around. In water and also in the air, also in the earth. You can have a lot. If you have small systems like you have, micro and nanoplastics are more dangerous
Yes.
than for bigger systems. For example, for us, we hope that it's not so dangerous than for smaller species. So I don't know if you have some information about micro and nanoplastics in these systems, the effect.
So what I remember now, we can search more about this, is two things. How we match the microplastics with the theory is that they are new vectors that are in this flux. So, and there are also studies that say that microplastics, at least in the ocean, somehow they carry a lot of pathogens. Pathogens like to stick there and grow there. So we have a new vector transporting microbes, and this vector also promotes the pathogens. So this is like a problem of having the plastics in all around. This is, for example, what now comes to my mind. But yes, there are many things that are related to this. Yes.
Mireia, I would like to know, do you consider in your conceptual frame also the interactions between microorganisms, for example, control by virus or something like this in the microbial communities, also how this can be affect the dispersion and especially the success of the succession?
You mean in between kingdoms, like, for example, virus? Yes, of course. If this is how has imbalanced the communities and this disturbed, then all the communities will be disturbed. For example, if we add a new species of bacteria in a community of bacteria that was not there before, will be a problem. This is, for example, now I'm thinking about those microbes that are transported, for example, from vessels, from one ocean to another. And those microbes, sometimes they are pathogenic. So when they arrive to new waters, they can cause toxic blooms that were never before there. And of course, this dynamic changes the whole community. It's not that we have this particular problem of this microbe producing toxins and blooms. This microbe also changes all, for example, the microbial loop in these waters where they are introduced. This is like an example.
And another question, so you say that the dispersion is not limited, but are you sure that we have not to consider stochasticity,
Yes.
especially in the success of the dispersion?
Yes, this is included in the theory, but I want to focus in this part that I'm sure it's like selected. But of course, the stochasticity of the environment or the system is there. This is part of all the dynamics. Yes, and now I was thinking that what, as you said, when a microbe enters to a new system, we can consider this as an invasive microbe. So we know the invasive organisms has many implications for the ecosystem, so this will be quite similar to others.
Okay, thank you.
Hello. If I understand correctly, you you can have different cycles that not necessarily are connected, like the deep sea to the surface, or this related to the air. I was just thinking, considering this cycle of the deep sea with the microbes, you're thinking about the photosynthetic microbes that can go to the deep sea. Have you ever think about like the other way around, like chemosynthetic bacteria that is from the deep sea and could take like the same path, but in the opposite way, like with the upwelling, they will be in the dormancy and then going back.
Yes, this is also contemplated in the theory. This is like an example, like the most simple I think we can think about the photosynthetic. But as you said, in the deep ocean is happening many things. And there are papers that say that those microbes from the thermal, I don't know now the name.
The thermal vents.
The thermal vents, you can, of course, they are more abundant in the deep oceans, but you can detect them in the surface. Yes, and
they are dormant in the.
Yes, they are dormant. So there is an evidence that says that yes, this is circulating. Yes. And in this particular work, they did, I think, a population genetics of these microbes from the thermal vents. So it's quite, it really supports this.
Hi, Mireia. Okay, hi, Mireia. And how is the scientific community responding to your theory? Have you received published comments or criticisms to the theory? I know it's too soon, but how is responding?
This is interesting because I learn a lot of how scientists and the information in scientists is spread and reside from the scientists. So far, I always receive positive comments. I also received emails from people that I never been in contact with, telling me this is interesting, this is linked with my works, blah, blah, blah. And also very papers that are published in very high impact journals that are citing this our paper and saying that this really match with what they find. So this is one part I think is quite positive. But personally, I think that maybe, I don't know what, I think this is quite personal, but if maybe I were another person from another country in another position, maybe this theory was more spread and more included or understood for a bigger community. But still, I think it has a very huge impact internationally.
Thank you, Mireia, for the talk. I really like it very much. And not only because of the nice picture of villages in the beginning.
Yes. Yes.
Ahora? Sí? So thank you for the talk, Mireia. I like it very much, and not only because of the nice picture of villages in the beginning, Altean Calpe, but also because the talk is really inspiring. I'm thinking of bigger animals, bigger individuals, bigger species. I was working on dispersal with lizards, and then I was reading a lot, and I really liked to see that you also have functional traits for dispersal in microbiome. And we work on dispersal in lizards, we look for the benefits of dispersal.
Yeah.
And are there benefits of dispersal in these microbial communities?
Yes.
Because there are functional traits. So is there any benefit? Of course, they disperse because of stochastic processes.
I think this is a difficult question. I should think more and elaborate this. But the benefits, how we can see it. A ver. So why they do this, right? This is like the question. Why they move? Why they don't stay? I think it's because this is quite linked with the living cycle. This is the first. So maybe if they are here always, they will have problems, like it's too crowded, for example. Maybe they need, this is very interesting. I remember a work that says that if you take samples, was also related with the sea, because my background is with the seawater. There is phytoplankton that it doesn't do this loop, it doesn't activate. So when they become dormant here, if they don't do this loop in the coast side, this is a work in Brazil. If they don't do this, they never activate again. They somehow need to be in contact with this unsuitable environment. There is something here that helps them to complete the cycle. And I think this is very interesting also. Yes, but yes.
Yeah. There are no more questions, so thanks a lot, Mireia.
Thank you, awesome. ¿Te gustó? Sí, ¿verdad?